Welcome everyone in this NPTEL online certification course on biological process design for wastewater treatment. So, in the previous last two lectures we studied regarding the overall stoichiometry of the two types of reactions that happen in the biological systems, and these reactions were anabolic and catabolic reactions that we have studied in detail in the previous two lectures. And we found that there are many reactions which happen under different conditions with different types of microorganisms for both anabolic and catabolic reactions. So, is it possible to combine them together and write an overall growth stoichiometry for the overall balance of the reactions? So, is it possible to combine them together? So, that is the what we are going to learn, and how we are going to combine them together. Generally, it is not possible to write an overall stoichiometry for microbial growth purely based upon the elemental balances and or oxidation reduction reactions that we have been writing for anabolic and catabolic reactions. The reason behind is that the much of the energy which is generated in the catabolic reaction that actually we studied earlier, it is not possible to determine that how much of that energy has been actually been transferred to the system, and energy transferred from the catabolism to anabolism via ATP and ADP is difficult to predict on the paper, because the amount of ATP that can be generated per unit energy generated by the catabolic reactions is difficult to predict. So, in general we cannot predict beforehand, or we cannot write that how much the energy which is generated via catabolic reactions have been actually been used by the anabolism reaction. So, it is very difficult to predict. Also, even for the same substrate and products and for the same growth conditions, the efficiency of oxidative phosphorylation reactions varies greatly among different microorganisms, and thus it is not possible in general to write an overall growth stoichiometry. But how to go ahead further? There is some conditions that mathematical clarifications will be given in this slide. We can see here that consider an overall growth stoichiometry of anaerobic heterotrophic microorganisms growing on a substrate of the general formula CXHYOZNZ. So, CWXY, actually this is CWHXOYNZ, and the overall growth reaction will be combination of both anabolic and catabolic reactions. So, therefore, the reactions will be based upon the substrate oxygen ammonia, while the products will be biomass carbon dioxide and water. So, this will be the overall growth reaction under aerobic heterotrophic microorganism when they are working. Now, the reaction can be written like this CW, and here we can see that the stoichiometric coefficient A, B, C, D and E are written with respect to oxygen, ammonia, biomass, CO2 and H2O. The above equation cannot be solved without any additional information, since there are 4 equations we can write, because the number of elements which are present in these reactions are carbon, nitrogen, oxygen and hydrogen. So, thus we can write 4 equations for 4 elements. Now, the unknowns are 5, A, B, C, D and E. So, there are 5 unknowns. So, mathematically also it is not possible to determine the value of A, B, C, D and E. So, what is the go ahead, what is the next step, how we can combine together and how we can find out the value of A, B, C, D and E. So, the additional equations are required to be able to write the stoichiometry of microbial growth, and that is obtained from the experimental data and is generally represented by growth yield. So, what we do is that we try to find out another parameter which is called as growth yield, and which is experimentally found out, and from this growth yield the parameters A, B, C, D and E, the stoichiometric coefficient are found out. The growth yield is defined as the amount of biomass formed per unit amount of substrate consumed. So, it is represented like this, where x represents the amount of biomass formed and S is the substrate. So, kg of biomass formed per kg of substrate, we can write biomass produced due to the growth and substrate removed due to the growth. So, this is there. The growth yield coefficient is usually obtained from the experimental data. If the growth yield is known, so if we can find out the growth yield, this constitutes an additional equation, and thus we have now 4 elemental equation, 1 growth yield. So, we have 5 equation, and thus we can find out the values of coefficient A, B, C, D in terms of the growth yield and the other parameters. So, this is how we can relate. So, for example, the example is given here. For aerobic metabolism, the reaction which was given earlier with respect to CW, HX, OY, NZ as substrate is shown here, and we can see that the A, B, C, D values are there. Now, we have 4 equations we can write based upon the growth yield. So, growth yield is written here, and here the growth yield will be defined as the amount of biomass formed. So, amount of biomass formed is C into molecular weight of biomass, okay, and divided by the molecular weight of substrate, because C is the amount of biomass which has been formed per unit amount of substrate, and here since the coefficient is 1, so it is not written here. So, this is C into molecular weight of biomass divided by molecular weight of substrate. So, this is the growth yield. Now, there are 4 balances we can write here. So, we can write carbon, oxygen, hydrogen and nitrogen balance. For carbon balance, we can see here that we have 5 C which is here plus D, the carbon is going here in CO2, and which is equal to W, which is because all the carbon is within the substrate, it is only the W amount is there. So, we have this carbon balance. Similarly, we can write for oxygen, hydrogen and nitrogen the way it is written here, and we can easily write this equation. Now, within this equation there is a one thing that this all the values are known. Now, what we do going further, what we do is that we solve by substitution, and for substitution what we do is that we write C in terms of y axis. So, the C value is written in terms of y axis into molecular weight of substrate divided by molecular weight of biomass. So, in this equation we can easily write for C, and this C is further used, C is equal to y xs into molecular weight of substrate divided by molecular weight of biomass. So, the C value is known to us, and using all other parametric values then can be found out using this, and the values of A, B, C can be found out in terms of W, X, Y, Z and the Y axis. So, this is what is written. We can find out the all the A value is written in terms of W, X, Y, Z and the Y axis, which is the growth yield. So, we can substitute all places and we can write this equation. So, it is evident from this equation that once the growth yield y x by s is known to us, the stoichiometry of microbial growth is fully defined and can be calculated with elemental balances as described in the previous slide. So, for the another case, if we take substrate is suppose glucose. So, in place of a generic substrate, if we write for glucose which is C6H12O6. So, if we write the balance equation for this, and if suppose the growth yield which is experimentally determined, it is 0.3 kg biomass per kg glucose, and it is a reasonable value for aerobic growth on many organic substrates. So, this value has been the yield has been taken as 0.3, and if we take the value of this 0.3 and glucose, thus we know the value of W, X and Y. So, in this case the W value is 6, the X value is 12, the Y value is 6 and the Z value is 0, and the ys value has been taken as 0.3. So, if we take all these values together and substitute in this particular equation, we can easily write this particular equation. So, thus if the growth yield is known, the substrate formula is known, we can easily write the balance equation. So, overall we can combine together, we can write the overall growth stoichiometric equations if the growth yield is known beforehand. So, this is the keyword. Now, for heterotrophs growing on organic substrate using nitrate as an electron acceptor. So, in this case already we have studied regarding the catabolic reaction and anabolism reaction. So, we can write for this, the reaction will be written here, it will be HNO3 and ammonia. In place of oxygen we are using HNO3 and overall we can write. So, here we have, in this case we again introduce, we have 6 coefficient A, B, C, D, E and F. So, now we have 6 unknown coefficient due to presence of molecular nitrogen which is coming into picture, because in the case of heterotrophs growing on organic substrates which use nitrate as an electron acceptor, nitrogen gets formed. Now, the sixth equation can be easily obtained by noticing some peculiarity in this equation, that the molecular nitrogen comes from the reduction of nitrate, and that all the nitrate that reacts is converted into nitrogen. So, this is the peculiarity, and from this what we can write is that the F value is always equal to A by 2. So, this F is actually we can always write in place of F here A by 2. So, in actual condition we have only 5 unknowns and 4 elemental balances we can write. So, thus using the growth coefficient again the growth yield, we can write the overall balance. So, overall growth of the heterotrophic microorganism on a carbon source using nitrate as an electron acceptor can be written as shown in the later table. Now, there is another condition that we have nitrifying microorganism. For nitrifying microorganism, combining the anabolic and catabolic reactions, the general growth equation will have the form of this. So, again we have 5 unknowns A, B, C, D and E. In this case we can see that CO2 is being used along with the oxygen for nitrifying microorganism, and ammonia is the base substrate. For nitrifiers, the growth yield is usually expressed as biomass produced per unit mass of nitrate as nitrogen produced. So, there is some difference in the growth yield which is given here. So, kg biomass produced per kg nitrate which has been produced. So, both in terms of production, and this equation can be written as introducing the molecular weights of biomass and nitrogen, and with reference to the reaction we can easily find that the value of this YXA upon NO3 it will be equal to C by D into 8.07. So, we can, after solving we can always get this particular equation. So, combining equation with the elemental balance of CHO and N and refer to reaction equation, this particular reaction equation, the overall growth can be written and it will be shown later on in the table. Similarly, for growth under anaerobic condition. So, here the same approach can be used to derive the stoichiometry of microbial growth under anaerobic condition. So, for fermentive bacteria the growth yield is always expressed exactly in the same way as for heterotrophic microorganism. So, this is similar to that, due to which the wide range of substrates and products that can be formed under fermentative condition, it is more practical to refer to particular fermentation reactions with reference to like conversion of glucose to acetic acid and hydrogen. So, if we case for example this particular equation, so the equation can be written here, the glucose is there and we can see that it is combining with water and ammonia to form the biomass plus acetic acid. So, we are referring only where the conversion of glucose to acetic acid, biomass and hydrogen is taking place along with the carbon dioxide. So, we have A, B, C, D, E and F. So, we have 6 unknowns, and 6 coefficients that have to be found out. Now, again we can write only 4 elemental balance, but additional equation is that the value of D will always be equal to del value of F, which comes from the fact that for glucose fermentation to acetic acid, carbon dioxide is the only produced in the catabolic reactions, which produces 1 mole of CO2 per mole of acetic acid. So, and because of this known fact, the value of D and F are equal. So, we can write here in place of F the value of D. So, thus we have only 5 unknowns and 4 elemental equations, and again we can use the growth yield definition to solve the problem. So, the equation can be solved usual to give this particular equation, and which is written here in terms of growth yield. This is written, we can solve, we can already have found out, and all the value can be found out if the value of y x by s is known to us. This represents the overall growth chemistry for the fermentative microorganism which convert glucose to acetic acid and hydrogen. So, we are referring to a particular equation only. Again, for acetoclastic methanogens, the general form of the growth stoichiometry is here. So, here we are referring to a case where the acetic acid is getting converted. So, acetic acid is combining with ammonia to form the microorganism plus methane, CO2 and H2O. Now, the growth yield is again defined in the usual way in terms of kg biomass produced per kg of acetic acid. So, this is the usual definition, and combining this we can write the overall equation in the form of this, and we can always find out the exact value of the coefficient once the growth yield is experimentally determined. So, this is the how we go ahead in solving different types of stoichiometric overall growth chemistry equation. Now, for hydrogenetropic methanogens, in which the hydrogen combines with carbon dioxide and ammonia to form the biomass plus H2O and methane. So, for this the growth yield is defined as kg of biomass produced per kg of hydrogen, and if we actually find out, the value will be, if we just write the molecular weights of biomass and hydrogen. So, it will be 56.5 C. So, this is the y axis by definition. So, we can see here the C value will be there, the molecular weight of biomass will be used and molecular weight of hydrogen will be used. And using this particular equation and the elemental balances, we can write the actual value of stoichiometric coefficient for each of the cases, and which can be solved once the value of yield coefficient is determined. So, this is how we can write for different equation, and the summary of the microbial growth reaction as the function of growth yield for various types of microorganisms can be written here. So, we have already seen for heterotroph aerobic reaction, heterotroph anoxic reaction, the growth yield is defined exactly in the same manner, and this is how the equations are written. So, we have previously seen these equations. In these two reactions, the difference is with respect to oxygen and HNO3 used, and then we studied regarding the nitrifiers, where the growth yield was defined like this: kg of biomass produced per kg of nitrate nitrogen, and then again using the yield coefficient we can write this particular balance equation. Similarly, for glucose conversion to acetic acid, the methanogens where acetoclastic conditions were there, and similarly for methanogens where hydrogenetropic conditions are there. For this, the growth yields are little bit defined differently: kg biomass per kg glucose for fermentative, kg biomass per kg acetic acid for acetoclastic, kg biomass per kg hydrogen for methanogens. And so, there is a difference in the growth yield definition, but using these growth yield definitions we can write the overall balance which is given here. So, thus using overall the growth yield definitions we can write the overall growth stoichiometry under various microorganisms operation. So, this can be done. Now, there is another thing, parameter or another important information that we can gather from these equations is the upper limit of growth yield. So, can you find, we have written so many equations. Now, can we find out the upper limit of growth yield using these equations? Yes, it is possible. So, from the overall growth stoichiometry, we can calculate some upper limits on the value of growth yield. For example, for metabolism to occur and for both anabolic and catabolic reactions need to take place, under that condition the higher limit of y x by s is the one in which no catabolic reactions have occurs. For example, for aerobic heterotropic metabolism of a carbon source, the reaction can be written like this. So, already we have written earlier the equation. Now, from this equation, in order for catabolisms to occur, the stoichiometric coefficient for oxygen needs to be higher than 0. So, that means that using this equation for oxygen, this is the stoichiometric coefficient. Now, for this reaction to occur, at least this coefficient value should be greater than 0. So, this is the condition that we apply. So, overall this equation is written here. Now, if we solve this, actually the equation can further be written as the y axis value will always be less than this particular value. Now, for example, if the substrate is glucose, so we have all the values are known to us. So, this condition means that overall we can solve this particular value by substituting different values. So, we can substitute for W is equal to 6, this x is equal to 12, y is equal to 6 and z is equal to 0. So, if we substitute in this particular equation and write the molecular weight of there is some mistake, this is substrate by biomass. So, this will be biomass by substrate. So, under this condition we can find out the value of y axis less than some value, and that some value is 0.75 kg biomass per kg glucose. So, that means the upper limit of growth yield for this particular reaction is 0.75. Actually, it will be much lower, and we have earlier taken the value to be 0.3, which was very reasonable. So, for the growth yield of, similarly we can find out for other types of reactions, like for the growth of hydrogenotropic methanogens, the need of catabolisms to occur means that some methane needs to be produced. So, that means the coefficient, this coefficient should be greater than 0. Now, if you solve it, actually it will come out that it should be less than 5.68. So, it is important to observe that the upper limit of growth yield calculated in this way refers only to the chemistry of the reaction and not necessarily to the energetics. So, in practical, the actual value of the growth yield will be much lower than the upper limit calculated here. However, it is always important to make a simple consistency to check whether the experimental data is correct or not, and the value of y axis is not exceeded. So, sometimes by mistake we can report such. So, as such we can find out for each of the reaction what is the upper limit of the growth yield using the method which has been defined here. Now, we will take some examples before closing today's lecture. So, this is one example in which it is being told that consider the anaerobic metabolism of amino acid glycine. One possible metabolism of this species under anaerobic condition is its conversion to acetic acid. So, we are trying to convert amino acid glycine to acetic acid. The microorganisms that carry out this reaction use glycine, ammonia and hydrogen. So, that means on the reactant side we have glycine, then ammonia and then hydrogen, and they produce new microorganisms, acetic acid and carbon dioxide and water. We know from biochemistry that hydrogen and acetic acid are only involved in the catabolic reactions, and that 1 mole of hydrogen is consumed per mole of acetic acid produced. So, already we have written, assuming that the growth yield is defined as kg of biomass produced per kg of glycine consumed, write the overall growth stoichiometry of anaerobic microbial growth on glycine. So, this is what we have to do. So, using the information which is given on in this example, we can easily write this particular equation. So, we have glycine which is reacting with ammonia and hydrogen to form the biomass, acetic acid, CO2 and H2O. So, we have 6 coefficient and we have the this y axis is defined. So, this will be C upon 1 into molecular weight of biomass into divided by molecular weight of glycine. So, we can easily calculate the molecular weight of biomass and glycine because their formula is given. Using this we can find out that okay, the y axis value is equal to 1.51 C. Now, similarly, the value of this for different C, O, H and N balance equations are given here, and already it is written that another condition is that 1 mole of hydrogen is consumed per 1 mole of acetic acid produced. So, this is another condition which is given here. So, using these conditions we can write 1 mole of hydrogen is consumed per mole of acetic acid produced, or that means we have B is equal to D, which is this is B and this is D. So, there is another condition that is coming here is B is equal to D. So, using this condition, molecular weight of biomass and molecular weight of glycine, we can easily write the equation and overall stoichiometry can be written by this particular equation which is given here. So, this is one example. Another example we can take for the anaerobic metabolism of palmitic acid, which is given by the formula C16H32O2. So, we have to find out for anaerobic metabolism. So, under this condition the palmitic acid is assumed to be converted into acetic acid and hydrogen. The overall growth reaction involves the consumption of ammonia, water along with the palmitic acid and production of microorganism, acetic acid, hydrogen and carbon dioxide. And we know from biochemistry that acetic acid and hydrogen are only produced during catabolism of palmitic acid and are not produced or consumed during anabolic reactions, and that yield coefficient definition is this. So, write the overall stoichiometry of anaerobic metabolism of palmitic acid. So, we can write this equation. Here we can see that this palmitic acid with ammonia, H2O and CO2 they are reacting together to form the acetic acid, hydrogen along with the biomass. So, if we can easily write, and this will be the equation with respect to growth yield, and once this is known also we can easily see that the carbon balance will be given by this particular equation, and the nitrogen balance gives that A is equal to D. So, it is very easy, we can find out that A is equal to D. So, A will become equal to the y axis by 0.44, and using all these conditions and using the molecular weight of the biomass and the molecular weight of palmitic acid, we can solve the overall, we can find out the overall coefficient in terms of y axis, the growth yield, and the overall equation can be written like this. And once the experimentally we can determine the value of y axis, we can exactly write the stoichiometric coefficients of this particular reaction. So, this way we can write the overall growth chemistry under various conditions and thus we can find out the overall stoichiometry of the equations. We have used following references for preparing this slide. You can always refer to these books for further understanding the overall growth chemistry. In total we have understood the catabolic reactions, the anabolic reactions and the combination of both. So, all these reactions are highly helpful in determining the actual reactions which are happening inside the bioreactors or the wastewater treatment reactors if the wastewater contain any of the conditions or the microorganisms and the what type of substrates are in the wastewater itself. So, we will be using this equation later on for various uses, and using this we can find out the mechanism, we can try to find out the overall growth yield etcetera also. So, these equations are highly helpful in understanding the microbial degradation processes happening inside the reactor during the wastewater treatment. So, we will continue with further sections in the next lecture. Thank you very much.